Hello, my name is Hristov, and today I'll be talking about persistent homology filtrations, merge trees, and contour trees. Now, what I'll be talking about in particular is a different way to set up filtrations with persistent homology, and it's going to apply to a very specific kind of data that's most often used in the sciences. So, for example, when scientists run numerical simulations, say a numerical simulation of weather, and then you generate physical properties, such as, for example, temperature, air, humidity. Those physical properties take on the forms of scalar fields. So those are functions from usually from R3 or R4 if we include time to R. So they have a single valued parameter. So what I'm going to do in this presentation is go over a simple example of data sets that emerge from those kinds of applications. I will tell you how we can set up filtrations on them and how the persistent homology of those filtrations can be encapsulated in something called merge trees and contour trees. So let me begin by setting up my example. So as I mentioned, as I mentioned, in a scalar field, what is generated is a bunch of numbers, single valued numbers, and those numbers are usually placed on a regular grid, most often a cubic grid. In this example, I'll be using a two-dimensional grid to make it easier to visualize. So here, I have generated 9 numbers, and I have placed them on a 3x3 grid. Now, currently, those numbers are placed in isolated vertices. So the next thing I'm going to do is I'm going to interpolate the whole space in this square. The first step in that process is to interpolate the edges between the numbers. That's using linear interpolation. And then the next step is to use barycentric subdivision to also interpolate the faces that's in between the edges. Now, usually when people talk about persistent homology, they want to study the shape of a particular data set. So that's usually a high-dimensional data set that has a complicated shape, and they try to approximate it. Here, the case is actually different. Here, our data set has a very, very simple shape. It is a contractible volume in space. Usually, you can refer to it as a 2 or a 3 disk. So it's not interesting topologically at all. But what is interesting for people to study is the function defined on the data set, the scalar function. So instead of using persistent homology to study the shape of the data, we're going to use it to study the shape of the function. And the way we are going to do that is when scientists are interested in features which can be defined using supralevel set or sublevel set components. Now, what do I mean by that? By a supralevel set, I mean that we set a threshold. We set a number. In this case, that would be between 1 and 9. For example, 2.8. And then everything above that threshold, the connected components of that will be the features. That will be of the supralevel set. If you look at the sublevel set, that will be everything below and their connected components. In the filtration we construct, we will be only interested in the first homology. So that will be, the zeroth homology, which is connected components. So the way we're going to set up the filtration is not with the standard Vietoris-Rips complex, where we grow a ball around every vertex. We are going to use the actual geometry of our domain. What we're going to do is we're going to have 2 filtrations, one ascending and one descending filtration. In the ascending filtration, we're going to add each one of the vertices one at a time. So first we add 1, then we add 2, and so on. And every time you add a vertex, we're also going to add all other simplices which have lower value. In the descending filtration, we're going to do the opposite. We're going to add the vertices from 9 to 1. And once we add a vertex, we add all simplices with a higher function value. So here is how that is going to look. So first, we're going to start with the ascending filtration. So in the ascending filtration, First, we add the simplex 1, which is just one connected component. And so on the right, I have drawn the barcode diagram for this filtration. So at the first step, we have one connected component. In the second step, we add the vertex 2. But since they are not connected in the original mesh by any simplices of lower value, then now we have two connected components. So a second one is born in this step. In the third step, we add the simplex 3. And again, we have two connected components because 1 and 3 are connected. Then we add the simplex 4. We still have two connected components. And once we add the simplex 5, then the two components merge. And usually with persistent homology, you have to decide which one merges into the other. So one persists and the other one dies. Usually we consider the component that is born first to persist and the other one to die when it merges with it. So now the two connected components merge and they become one connected component. And throughout the rest of the filtration, we are only going to have this one connected component because every time we add a new vertex, it will already be connected to the previous ones. So as we continue adding vertices, eventually we construct the whole mesh, and at the end we have one connected component. So that was the ascending filtration. Now I'm going to demonstrate the descending filtration. So in the descending filtration, we are going to do the opposite. We're going to start from the top down. We're going to start by first adding the vertex 9. Then we add the vertex 8. Now we have two connected components. Then we add the vertex 7. We still have two connected components. And then we add the vertex 6. So now we have three connected components. And in this step, the third one is born. After which we add the vertex 5, we still have three connected components. And when we add the vertex 4, we have two connected components. If we continue on doing the same thing with the filtration, all of the components eventually merge into the same component. And that's, of course, because at the end of filtration, we get the whole data set, which is just one connected component. As I said, we apply this when our data set is intractable. It is a disk volume in space. So far, I have described how to construct a ascending filtration and a descending filtration. After we constructed those filtrations, we can, of course, run a persistent homology algorithm to compute the zeroth persistent homology. And we can visualize that using barcodes. So this would be the barcode for the ascending filtration, and this would be the barcode for the descending filtration. Now, the barcodes are a very good way of visualizing how the connected components of the superlevel sets and the sublevel sets merge. But the barcodes aren't that interactive. In order to actually use that information, it is often more convenient to represent those barcodes as trees or as graphs. So here's how we're going to do that. So for the ascending filtration, we're going to construct a tree which corresponds to how the components merge. The leaves of the tree would be where connected components are born, and the saddles of the tree are where connected components merge. Now, once we have the tree, the convenient thing is that we can use algorithms from computer science to traverse the tree and to extract information from it. For example, if we look at the descending filtration in the joint tree, we can use the tree to first identify a seed for each one of the connected components, so 6, 8, and 9, and then we can traverse those connected components given a threshold. Once we have done that, there's also another important thing to mention. Usually in persistent homology, the way we do the pairs is using the elder rule. So the component which is born first persists and the other one does not. With the split tree, we have a bit more freedom. So at each one of the saddles, we can make a decision which one persists and which one does not. And we usually in practice, it is not often the best to take the elder rule. Sometimes we may want to pick pairs by volume. So for example, if we look at the two connected components, one starting at 8, one starting at 9, both merge at 4, we can see that the one which is born at 8 would have more volume because it contains more vertices. So if we have the tree and we decompose the tree, we can actually use a different pairing for that. So in conclusion, in this talk, I have talked about how we can set up filtrations without using vector strips complexes. And those filtrations would work on simply connected domains where you have a regularly sampled grid, which is usually the output of scientific simulations. And then I have talked about how we can visualize and work with the output of the zero persistent homology using the split and the join tree as analogous to the ascending and descending filtration barcodes.